thank you for the organizers for having me today. And I'll be talking about a different virus, at this part, during this part of the conference. Now, norovirus has been found to be responsible for at least 50% of acute gastroenteritis cases and outbreaks worldwide. It's actually ranked second after rotavirus in being able to cause viral gastroenteritis across different age groups. And it is the leading cause of gastroenteritis seeking medical care in emergency departments, outpatient clinics, as well as the community. Now, 10 to 15% of the severe cases of acute gastroenteritis happen among children less than 5 years old, and the mild to moderate cases of diarrhea are actually found across other age groups. The virus is primarily transmitted through the fecal-oral route, and it has a short incubation period of 24 to 48 hours. And in most of the cases, the illness is self-limiting and resolves within 12 to 72 hours, with, of course, a few exceptions among young children as well as elderly and immunosuppressed patients. Now, the symptoms are similar to many other viruses causing acute gastroenteritis, so children as well as young adults and elderly would suffer from vomiting episodes, from diarrheal episodes, nausea, abdominal pain and cramps, as well as low-grade fever. Now, in most of the cases, the treatment actually involves the reversal of dehydration as well as electrolyte deficiency. The genome of the virus, which is norovirus, is actually a linear, single-stranded, positive sense RNA virus, and the genome is made up of three open reading frames, ORF1, 2, and 3. And the first open reading frame actually is the part encoding the non-structural proteins, which include the RNA-dependent RNA polymerase. And open reading frame 2 and 3 actually encode respectively the viral protein 1 and viral protein 2, VP1 being the major capsid protein of the virus, and VP2 is the minor capsid protein. Now, based on the amino acid sequence of the VP1, noroviruses have been classified into 6 genogroups, with genogroup 1 from 1 to 7, and genogroups 1, 2, and 4 actually are the one causing, causing diseases among humans. Now, the genogroups are further, subclassified or subtyped into genotypes based on the amino acid sequences of the RNA-dependent RNA polymerase. And this is why, the nomenclature would be G1-3, G2-4, and others. The most common genogroup and genotype of noroviruses causing epidemics as well as outbreaks worldwide is actually norovirus genogroup 2, genotype 4, or which I will be referring to as G2-4 across the presentation. Now, in Lebanon, and to our knowledge, we have no studies on norovirus and its association with gastroenteritis. And this is why the aim was to assess the association and the prevalence of norovirus, gastroenteritis, as well as its genotypic characterization among hospitalized children less than 5 years old. And we've collected samples, actually stool samples from more than 1,500 patients that less than 5 years old that presented with diarrhea to different hospitals in Lebanon. Actually, our samples were collected from 6 different hospitals, 3 from the capital Beirut, and 1 from one hospital in the northern part of Lebanon, and 2 hospitals from the southern part of Lebanon. As I said, the age group, the children were less than 5 years, and we've collected these samples from January 2011 to June 2013. And those samples that tested negative for rotavirus were claimed eligible for our study, and we've ended up with the 739 stool samples to be looked at for norovirus infection. We've isolated the viral RNA, we've extracted the viral RNA, and then we've performed RT-PCR, and then the PCR products were sequenced and phylogenetic tree analysis were inferred. And table 1 actually shows us the demographic, some of the demographic characteristics of our study participants by gender, age group, and region. Now, out of the 739 samples that we've collected from children less than 5 years old and hospitalized due to diarrhea and acute gastroenteritis symptoms, we've had only 83 samples, 83 children being positive for both norovirus genogroup 1 and norovirus genogroup 2. Now, which leads to so the 11.2% of our sample tested positive for norovirus, with 10.6 being positive for genogroup 2 and less than 1 for genogroup 1. When we looked at the distribution of male-female ratio among these children, so the majority of this group that tested positive for norovirus infection were males, and the majority were between 1 to 2 years old, and most of the cases were present in were collected from samples or from children in the northern part of Lebanon. Now, we did not find any significant difference, being like gender-wise or age group association with infection. The majority as well of the children that were hospitalized had the regular symptoms of norovirus infection, with 67% showing with fever, 77% showing with vomiting, 95% with diarrhea, and mild to and 77% showing mild to moderate dehydration. When we looked at the Vesicari score, which is the clinical scoring system for acute gastroenteritis, more than 85% of our cases were classified as severe according to the Vesicari score. Now, when we looked at the incidence per region, we tried to see whether there's a significant difference in terms of regional incidences. We found that, the incidences in Beirut was 9%, in the north 11%, and 13.6% in the south, and we did not find any statistical significance between these incidences. We also looked at the number of cases and when did they occur across time during the course of the study. And we found that the peak of our cases were actually occurring in July of 2011 and 2012, which is the summer season in Lebanon. Of course, we had sporadic cases that took place during the course of the year and during other months, but the clustering of our cases were found during the warm weather in Lebanon. Now, in order to assess the genetic diversity of the isolated cases of norovirus among our study participants, we inferred the phylogenetic tree of the major of a partial component of VP1, or the major capsid gene. And we found that, as I mentioned previously, we had 83 cases that tested norovirus positive. 5 of these were norovirus genogroup 1. And among these 5 cases, 4 of them were G1.3 and 1 G1.4, and then the rest of the cases were G2. And then within the G2 genogroup, we had 68% being G2.4. And then what ranked second after G2.4 was actually G2.6, 9%, and then the third were G2.3 and G2.21, and then we had a few cases of G2.9, 13, 1, and 2. Looking at the G24 phylogenetic analysis, we found that during 2011 we had viral variants circulating during that year that were similar to the Apeldoorn 2008 strain, which caused epidemics, outbreaks in the Netherlands in 2008. And between 2012 and 2013, we had our cases similar to the Sydney, Australia variant that also is a major cause of epidemics and outbreaks that were first identified in Australia in 2012 and then later on affected other countries. So in conclusion, our results are actually compatible with globally reported ones, whereby we found that G24 contributed to the majority of the viral gastroenteritis cases that we looked at in our sample. And we had a co-circulation of two different lineages of norovirus G24, the Apeldoorn one in 2011 and the Australia G24 Sydney strain between 2012 and 2013. And this is actually happening in the co-circulation of two lineages of norovirus is actually something that is happening in many countries. And it has been suggested that this is actually associated with selective pressure to generate new mutants of the virus. Now, G26 in our sample ranked as a second predominant cause of gastroenteritis among hospitalized children in Lebanon, which is similar to what has been reported in other countries, specifically Brazil, Japan, South Africa, and Finland. G23 and G221 ranked third among our study participants, while these have been reported to be second in other countries. Now, the seasonality of norovirus actually has been debatable across the literature, and in many reports and many modeling systems, it has been suggested that norovirus is actually a winter phenomenon, whereby the cases of norovirus happen during the cold months. Other reports which align with our results, whereby norovirus is happening in warm weather or during the summertime, and this is what we found in Lebanon. And however, more studies and more investigational research need to be done in Lebanon and the Middle East and North African region in order to really report on the seasonality pattern of norovirus and to make a real analogical or comparative analysis with the other parts of the world. We have few studies in the Middle East and North Africa looking at the association of norovirus with gastroenteritis. And with those studies, few of them actually have reported that G23 is being more prevalent. But as I said, there's a lack of studies on norovirus in the MENA region, and the lack of the data on the genetic relatedness and subtyping. But this is something that we are working on we are working on expanding in future years. So the variants of norovirus G24 lineage have been actually associated with 62 to 80% of outbreaks worldwide. However, norovirus remains an understudied causative agent of acute gastroenteritis, and actually norovirus infection is not included in the global burden of diseases among the infectious agent causing gastroenteritis and diarrhea. And this is probably actually affected by the lack of the use of clinical diagnostic methods to look for norovirus in many parts of the world, and specifically the developing countries. So it's clear that we need to further investigate this virus and further surveillance strategies in order to report it and associate it with acute gastroenteritis worldwide in order to support intervention strategies. And finally, I would like to thank my lab at the Faculty of Health Sciences, Khalil, Rana, and Noor, and my collaborators at the Centers for Infectious Disease Research at the American University of Beirut, Dr. Zbeibu and his team, and Dr. Zaraqit, and thank you very much.